So with that, today I wanted to put out a sort of call for not so much help, for involvement of my supporters. And I'm going to explain exactly what I mean by that, how I want you to get involved, and how I can best use your support. And one of the reasons for this is, as probably a lot of people know, there's a lot of talk these days about the quote no virus team, the no virus position, and we are being contacted, we being mostly Andy Kaufman and Mark Bailey and myself, to participate in events and quote debates and conversations and all kinds of things so that our position can be examined, they say. Now we are in negotiation for how we're going to do this, and I must say I have my doubts as to whether any of them will actually happen, but they may happen. And in any case, we're going to put out a statement, I think, which will outline our position. And as you'll see, I'm going to ask for your help in a bunch of claims I'm going to make. And I know some of you are sort of crack researchers and you love to go looking for studies and research. So I'm going to clearly say, here's a point where I see no study that demonstrates this. And then I'm going to have everybody or ask you to look. And if you can find a study or a paper that contradicts what I'm saying, I would love if you could send it to me or send it to our customer support, or even put it in the comments under this video. And that will help me make sure that I didn't miss anything. Obviously, I've tried to not miss anything, but you never know. And that brings me once again to the question of why is this so important that the world understands that there is no virus, and the science of that is basically somewhere between incorrect and maybe even fraudulent. Why do I need to go on this for the 1000th time? And let me just show you a few of the reasons why, because even though we are gaining in leaps and bounds, and that's why people are even knowing they have to engage with us, we still have a lot of work to be done. And I'm going to show you some examples of the work that needs to be done. So whoops, I didn't share the sound. Okay, so the first one, this is a guy who I sometimes take a look at. His name is Paul Craig Roberts. He used to be, I think, Assistant Secretary of the Treasury under Reagan, and has come to the point of disbelieving a lot of things that most conventional people believe. He talks about 9/11 as an inside job. We have the COVID pandemic as an inside job. He talks about a lot of these things. He talks about the real issues that are happening in the Ukraine, and keeps wondering why Putin and the Russians are making such bad decisions. And I would love to talk to him about that at some point, because I think I can explain exactly why that's happening. But the issue that I'm talking about today is, as people know, there was a recent congressional testimony, and here's what Paul Roberts said about this. Folks, this is a big deal. In a recent testimony, Robert Redfield, who was director of the Center for Disease Control during the COVID pandemic, told Congress that Dr. Fauci financed the gain-of-function research that made the virus so contagious. The virus was not natural and engineered in a lab, and that Fauci excluded him from meetings and decisions because he, Redfield, was disturbed about what was going on. There is no doubt of Fauci's guilt, but with Democrats controlling the White House and Senate, Fauci cannot be held accountable. The mass murderer is free, while Trump supporters who attended a rally are in prison. This is what a Democrat rule means. Americans who vote Democrat vote for a Third Reich regimen. So Mr. Roberts is always complaining about how duped and stupid the American people are. He uses a different word than that. And it's just ironic that after all his complaints about COVID being an inside job and the pandemic that wasn't, he would believe the former director of the Center for Disease Control saying that this, quote, virus was made so, quote, contagious because of gain of function research. And I guarantee that Mr. Roberts has no idea exactly what was done to make this, quote, virus so, quote, contagious, and could not explain to anybody that there is such a virus and why he would believe this Dr. Redfield and all the other people and the Republicans and anybody else who's claiming such a thing, when the reality is far worse, that all of these people were in on this hoax, this farce. And the real story, which apparently he's missing, is there is no virus. So the Democrats and the Republicans are both in on this, and they're putting a staged event together so that we can think there are two sides of this debate. There's the side that it's the natural virus, and there's the side that it's a gain of function by the bad Fauci. And if we just get rid of Fauci and put those mean people in jail who are doing horrible things, this would all be over. And the reality is this debate will go on forever, because the reason they can't decide whether there's a lab leaked or gain of function virus is because there is no virus to decide. It's like deciding whether the invisible flying explosive unicorn was engineered or not, and nobody remembers there is no unicorn. So I'd be happy to talk to Mr. Roberts about that, and he could learn the real story. If anybody can pass this to him, I'd be happy to have a private conversation so he could correct him and not end up being essentially fooled himself. And that gets to my next story, which is on the similar lines, which was in the somewhere. I don't remember where I got this from. It says the China announces new lockdowns for the flu, and Washington Post tells Americans to prepare for the same. And they say Chinese officials want to bring lockdowns to combat the flu, leaving many furious about a prospect of returning to strict restrictions seen during the COVID-19 outbreak. And the Mail has the story. And as they say, the Washington Post says, Americans beware, because we're going to bring this to you as well. So this is far from over. They're going to do this virus story again and again, and the only possible solution is for people to realize there is no such thing. And that brings me to the final clip, which probably many of you have seen.
Occurred to me, because I know while we're talking about gain of function, we're talking about lab leaks, there's this little group of people out there that keep talking about terrain theory and germ theory and this conversation that many of you may not even understand. But I just want to say this. We are going to get into those conversations further down the road with some of the people out there. But if what you want is to remove the idea that any of this ever existed, that any of this ever happened, then what you're saying is you want Tony Fauci to walk off scot-free. You want all these liars that lied to us and locked us down to get away with this by saying, oh, the germ never existed, it was never there, when clearly the science is turning on them. Their own science is turning on them. And right at the moment where we are going to move into indictments and take these people out, what some of you out there want to do is let's just change the conversation altogether and let these criminals that destroyed our lives walk off into the sunset. I don't understand it. I don't understand why that would be your conversation right now. For those of you that don't know what I'm talking about, just wait. We're going to have this conversation further down the road. All right.
Okay, just wait. We're going to get these bad guys. This reminds me of the comic Peanuts, where I think it was Lucy kept holding the football. And every time she said to Charlie Brown, who wanted nothing more in life but to kick the football, and every time he would come to run and kick the football, she would take the football away and fool him once again. So we were told that we were going to get Hillary Clinton and lock her up. And then we were told that as soon as Trump got elected, never mind. We've been told this story that if we just put the bad guys in jail over and over again. So we put the bad guy in jail and then we get what? Peter Hotez. And somehow that's going to save you and affect your life. And somehow by saying that this whole thing is based on a fraud, that somehow what we believe is that somehow what we think is to let all these guys off who've been lying to us about all this stuff, and we're about to get them, and don't we understand that? And that to me is absolutely total nonsense. What you will realize when you hear criticism, and they've rolled out the criticism of this is divisive, nobody wants to talk about this, nobody can understand, there's no germs, there's no viruses, nobody can understand the science, the people can't possibly understand this. Nobody actually talks about the science. They just roll out one excuse after another. First it was divisive, then we're trying to change the subject. Then we want the bad guys to walk free. Just at the time when the good guys are about to indict them based on the science, we're going to get them all. And how many times have we all heard this story? And it's a whole lot. And I can tell you it ain't going to happen. And if it did happen, it won't make any difference to your or my life, because the whole system will go on like nothing happened, just like it always does. So with that, now let's get into the part that I'm going to ask your help with. I'm actually going to read this. So this is essentially, I don't usually do this, but it's kind of the script that I'm going to open this virus debate about. And I don't usually do this. So this is a different way of explaining this, and then you will see where I'm asking for your help. So this is another way of explaining the history of virology. So here is the history of virology. They start by wondering if the observation that often people get similar symptoms at the same time means something was being passed between them. And we're talking now late 1800s, early 1900s. The scientists at that time, did proper contagion studies, like the Rossino study with the Spanish flu. You've all heard of that over and over again. And over found that being around sick people did not make well people sick. That should have been the end of virology, as well as the germ theory from which virology was invented. If you, our detractors, have a paper that shows that this is incorrect, please send it to me or us, or put it in the comments at the bottom, before the conversation that we have. Otherwise, we will use this and accept this as a fact. Let me read you some of the other synopses of some of the other studies that have been done to understand this common observation that well, my children or my golfing buddy or somebody was sick and then I got sick. Now, we've been over the example of, just being around somebody who's sick and then you get the same symptoms. First of all, we're not talking about viruses. We're talking about the phenomena of transmission, sorry. And so if you believe that, you believe that if sailors on a ship got sick, one after another, same symptoms, that somehow that illness we call scurvy is a contagious, transmissible illness. The same with the other example that I use of you have 100 rats in a basement, and unbeknownst to you, somebody puts some rat poison. The first day, 10 rats all bleed to death and die. Then the next day, 10 more, and so on. All the same symptoms, one after another. They even die. And so that if that proves transmission, same symptoms, temporally the same, and when, in other words, the same time frame, one after another or 10 after another gets sick, somehow that proves A, transmission, and B, it's a virus. But let's read some of the other experiments have been done. So in March of 1919, Rosano and Keegan conducted 9 separate experiments on 49 healthy men to prove contagion. In all experiments, 0 of the 49 men became sick after being exposed to the sick people or the bodily fluids of sick people. And here is the reference from JAMA. And then there's another reference from Rossino again. 0 of the 50 men got sick when exposed to the flu. And we've all heard about that. And then in 1919, Wall et al. Conducted 3 separate experiments to infect 6 healthy people with influenza by exposing them to mucus secretions and lung tissue from sick people. 0 out of the 6 men contracted influenza in any of the 3 studies, and here is the reference. In 1920, Schmidt et al. Conducted 2 controlled experiments exposing healthy people to bodily fluids of sick people. Of 196 people exposed to mucus secretions of sick people, 21, which is 10.7%, developed colds and 3 developed grip. That's the flu, 1.5%. In the second group of the 84 healthy people exposed to mucus secretions of sick people, 5 developed grip, 4 developed colds, that's 5.9 and 4.7%. Of 43 controls who had been inoculated with sterile physiological salt solution, 18.6% developed colds. In other words, even though it said, you might think, oh, well, almost 10% of the people got sick, exposed to the sick people. It turns out that 18% of the people who were exposed to essentially nothing, a higher percentage of people got sick after being exposed to saline compared to those who were exposed to the, quote, virus. This is a PubMed, and I have the number right here if anybody needs it. In 1921, Williams et al. Tried experimentally to infect 45 healthy people with the common cold and influenza by exposing them to mucus secretions from sick people. 0 of the 45 became ill. And here's another PubMed reference. In 1924, Robertson and Groves exposed 100 healthy individuals to bodily secretions from 16 people suffering from influenza. The authors concluded that 0 out of 100 became sick as a result of being exposed to the bodily secretions, and here's the reference. In 1930, Dochez et al. Attempted to infect a group of men experimentally with the common cold. The author stated in their results something that is nothing short of amazing. Quote, "It was very apparent very early that this individual is more or less unreliable from the start. It was possible to keep him in the dark regarding our procedure. He had inconspicuous symptoms after his test injection of both broth, sterile broth, and no more striking results from the cold filtrate until an assistant on the second day after injection inadvertently referred to this failure to contract a cold. That evening and night, the subject reported severe symptomology, including sneezing, cough, sore throat, and stuffiness in the nose. The next morning, he was told that he had been misinformed in regard to the nature of the filtrate, and his symptoms subsided within the hour. It is important to note that there was an entire absence of other pathological changes, which only goes to show that one of the main risk factors for supposed transmission of an illness, as in playing golf with somebody and getting sick, turns out to be the belief that such a thing will happen. In 1937, Burnett and Lush conducted an experiment exposing 200 healthy people to bodily secretions from people infected with influenza. 0 out of 200 became sick. And here is the PubMed reference. In 1940, Burnett and Foley tried to experimentally infect 15 university students with influenza. The authors concluded their experiment was a failure. Nobody got sick. Here is another reference. I can give anybody the reference if they need them. This was from an article called The Contagion Fairy Tale. Contagion, a Fairy Story. So here is my request to all of you. If anybody out there can find a study that contradicts this, which was done properly, showing that illnesses which we say are viral or contagious actually make well people sick when the exposure is in the so-called normal way, like drinking it or spraying it or exposure to another person, please send that to me as soon as you can find it so I can correct this. As far as we can tell, that study doesn't exist. Sorry. Now, number 2, this should have been the end of virology. In other words, there is nothing transmissible. Therefore, why look for an agent of disease in the secretions or fluids of somebody who's sick? But it wasn't the end of virology. So they then decided to look in sick people's tissues and secretions for a pathogen. They found bacteria and fungi, but nothing they could call a virus. They then filtered these secretions and ground up tissues and passed this ground tissue through a filter that removed bacteria, fungus, dead cells, etc. This filtrate would have contained the virus if present. They exposed animals to this filtrate. None got sick, except if they did this exposure in a highly abnormal and uncontrolled way, such as intubating the animal and stuffing this filtrate fluid or the supernatant, if they centrifuged it, down into the lungs of these unfortunate animals, and then seeing if they got sick. And again, sometimes they actually got a cough, which isn't surprising. They almost never, in fact, never that we have seen, have done proper controls. They sometimes injected this filtrate or supernatant directly into the animal's brains or into their eyes, or other crazy ways of supposedly transmitting illness. And apart from that, none of them got sick. This should have been the end of virology. In other words, we now know as a fact that the filtrate and supernatant from people who are said to have a viral illness, when that filtrate or supernatant is exposed to the people who are well in a normal way, as far as we can tell, there is no paper that shows that those well animals or people got sick. Now, if anybody can send me a paper that shows otherwise, I would love to see it. So far, from the people we're trying to negotiate with, we have no papers even addressing this issue. Therefore, I will take it as a fact. Okay, number 3. Then, and now we're talking 1930s or so, They invented a microscope called an electron microscope. Later, they call it a transmission electron microscope, that they thought they could see particles in the filtrate of sick people. Now, if you think about this logically and reasonably, why would you even look for anything in the filtrate or supernatant, that's the liquid part after you centrifuge it, of sick people, if you already have proven that the filtrate doesn't make any animal or person who's well sick? What would be the point of looking in there? We already know there's nothing in there that causes illness. But anyways, they did. But like I say, this should have been an irrelevant finding, as they already had shown that the filtrate didn't make anybody sick. They found such particles. Later, they found out that these particles are not exogenous from the outside, disease-causing viruses, but simply the person's tissues breaking down in the usual manner, meaning the usual manner that tissue breaks down when you're sick. This should have been the end of virology, but it wasn't. If anybody out there has a paper proving that these particles seen on any kind of electron microscopy are purified exogenous viruses that are then said and proven to make anybody or any animal sick, please send that to us before the conversation. Otherwise, this statement stands as fact, and many of you will remember that I have presented a number of studies showing that we now know with SARS-CoV-2 and many other viruses, HIV, that these electron microscope pictures of whatever sort have in no way been proven to A, be a virus, B, not come from the breakdown of the tissues, C, contain the genetic material that they say the virus contains, or the proteins they say the virus contains. These pictures are totally disconnected from all of those other findings, and there is not an example that we found of a purified, isolated, so-called pathogenic virus that has been documented by electron microscopy of any sort. If anybody has a paper that proves me wrong, I would love to see it, and send me the paper or the reference as soon as possible, because as far as we've put this out, nobody has been able to do that. Therefore, this stands as fact. So, number 4, then, in spite of having already shown that the filtrate or supernatant of a sick person's fluid doesn't make anyone sick, they decided to prove that this was incorrect. The technique they chose to do this was to take this filtrate or supernatant and inoculate, spread that on cell cultures. In the early days, they ran what would be approximate controls and showed that it wasn't any pathogen in the filtrate that was killing the cells, but the experimental procedure itself. We repeated this experiment in the last few years, and it's been repeated also a number of times, especially in the early days of the cell culture, and again demonstrated that the experimental conditions, not any assumed virus in the sample, is that kills the culture cells. This is what virologists call isolation. So it stands as a fact that no virus has ever been isolated and therefore shown to exist. If you disagree, please show us a paper that proves the cell culture experiment is not only a valid scientific way to demonstrate the existence of a virus, but that it has been done with proper controls. Since we have found no such paper, our statement that a virus has never been isolated, meaning it cannot be said to exist or be further characterized, stands as a fact. I also want to point out that all of these 4 points are internally consistent and tell the same story. In other words, sick people don't make well people sick. So then they look, is there something in the sick person, the chickenpox blister, the mucus, the lung fluid that makes well people sick? Now, if we've already shown that it's the person as a whole doesn't, why would you look? But they did look, and no surprise, they show that it didn't work. Then they said, okay, we're going to look in those fluids and see if we find some organism, some particle that would be making these people sick. Why would you do that when you've already shown that the sick people and the part of the sick people doesn't make them sick? Why would you look for something in there that makes them sick? But no surprise, again, consistent with the whole narrative, they can't find anything in those fluids that they can show is a so-called exogenous pathogenic virus. And then, even though they've shown that, they decide to take that sick fluid, the liquid from the sick person's fluid, and inoculate that on cell cultures to see if it makes the cell cultures sick. Again, why would you do that if you've already shown there's nothing in there and essentially the entire product doesn't make anybody sick? But they did do it. And again, no surprise and consistent with the whole narrative, they show that there is no particle, there is nothing in that supernatant that's making those cell cultures sick and die. So the whole thing lines up perfectly consistent as you would expect. But then, in spite of all reason or logic, they decide to look in this supernatant to see if they can find a genome. And so, finally, current virologists are somehow claiming that in spite of the fact that no virus has ever been demonstrated to exist in the tissues or secretions of any sick person, they want us to believe that they can find the genome of this particle in exactly this fluid. Besides the fact that this strains logic and credulity, a farewell to virology shows conclusively that no such genome has ever been scientifically validated. Therefore, unless we are shown a paper to the contrary, this too stands as fact. The important point here is that there was never any reason to engage in step 5 in the first place. After showing that the steps 1 through 4 demonstrate there is no evidence that a particle called a virus even existed, and there was not and never will be any sense or reason to go looking for a genome of this particle. And again, if anybody has a valid, scientifically validated study showing this genome must have come from this particle, which again has never been found. So if you can find that this particle has been found or has been proven to kill cell cultures, please send us that study and we will correct ourselves. So far, we haven't received any such studies because as far as we can tell, these studies do not exist. And therefore, virology stands as a disproven hypothesis which should be relegated to the dustbin of history. So again, what am I asking for? I'm asking for, 1, a contagion study that shows that sick people exposed to well people make the well people sick, done in a proper scientific way. Then we're asking for a study that shows that any part of the sick people, like a blister or mucus or bronchial fluid, makes the well people sick when done in a scientific, controlled, normal way, not injected into their lungs or their brain or their eyeballs. Then we want to see a study proving that any picture, photograph from any kind of electron microscopy paper we have proven is an exogenous pathogenic virus, because we've never seen that. And then that the cell culture experiment proves that it was the virus and only the virus alone in the filtrate or supernatant, which could have caused the cell death, the cytopathic effect, and not something else in that fluid and not the conditions of the experiment. And then finally, a study showing that a genome of a particle which has at this point not been found, somehow we know that genome came only from that particle, and the whole thing strains logic and credulity and is completely irrational as far as I can see. And again, I'm asking for anybody's help to show me that any of these studies exist, because this is one of the things we're going with in these potential upcoming debates.